Hello, everybody. Today we are going to talk about the analysis of PHD3 and its effects on HIF-alpha subunits in a hypoxic environment. This presentation is by me, Kerollos, Roufael, and Stephen Galvez. So as an abstract for our presentation covers the function of PHD3, how it is related to multiple myeloma, which is a disease, an incurable disease that affects the bone marrow that can cause cancer. Also, some ways that we can prevent the growth of PHD3 and how it is related to HIF-alpha, and some techniques as Western and immunoplots. Through them, how we can figure the way that PHD3 works. Finally, we compared the trypsin to PHD3, and we figured its relation to metastasis. And at the end, we have our references. So at first, I would like to talk or give a quick introduction of what's PHD3 and its role in cancer. So PHD3 is a tumor suppressor gene, which basically promotes the neural apoptosis in response to hypoxia, which basically means low oxygen, to regulate HIF-alpha. And also it helps in induction of sensitivity of myeloma cells. PHD3 hydroxylate the HIF subunits on carbon 4 of proline, which residues it and causes it to lysis in normaxic conditions. Under hypoxic conditions, we will see the expression of PHD3, which allows for interaction of HIF-alpha, which can lead into apoptosis of multiple myeloma cells. Finally, if expressed using lentivirus vector or by overcoming its epigenetic silencing using dimethyl transferase inhibitor, then we can prevent the multiple myeloma from engrafting.
All right, so we used a Western or an immunoblot to demonstrate PHD3's interaction with HIF-alpha. And the drawing shows two different Westerns, one showing normoxic conditions and one showing hypoxic conditions, where the multiple myeloma cell was cultured over 48 hours in said conditions. And then we lysated the cell and we ran the lysate through SDS page. And from there, we used a antibody once the gel was moved into a membrane. And then we use an antibody to precipitate either HIF-alpha in hypoxic conditions or PHD3 in normoxic conditions. And you could see that under normoxic conditions, PHD3 is present, which when PHD3 is present degrades HIF-alpha, and the opposite occurs during hypoxic conditions. You can move it to the next one. And here is just a more detailed explanation. But basically, I just summarized what was described or what was drawn in the previous slide. So PHD3 is related to trypsin in the fact that they both work metastatically. PHD3 interacts with HIF by hydroxylating the proline residues, as we said before. And this leads to ultimately the degradation of HIF through a few different processes, ubiquitination and other details that we won't discuss here. But basically, hydroxylation causes HIF to degrade, and because of that, HIF cannot function, and HIF's function in hypoxic conditions will cause angiogenic genes to be expressed and translated, and those genes will cause genes such as genes that code or help with anaerobic respiration, or genes that will expand the vascular system within cells to get more oxygen, will be expressed, and that is described as angiogenic. So if HIF is angiogenic, then we can assume that PHD3 is metastatic. And metastatic is associated with trypsin. So the properties are very similar, or are similar to trypsin in that sense. And that concludes our presentation. And our references are made here on the final slide. Thank you.
Okay, I'll